mechanisms that link nutrition and toxicology with the goal of developing translatable prevention strategies to inflammation-induced metabolic disorders, including preterm birth, obesity, as well as cardiovascular disease, especially in at-risk populations. This current NIEHS funding includes an R01 investigating how PFAS exposure accelerates atherosclerosis in adults, and a second R01 examining the links between maternal PFAS exposure and placental toxicity and preterm birth. He's been recognized by NIEHS's Stories of Success series for his work on how environmental pollutants and nutrition interact to shape health outcomes. And I'm happy to turn it over to him.
Thank you, Matt. Thank you, everybody, for the introduction. And Anna, thanks for the invite. Matt, you guys are great. Carolina, thanks for organizing everything. So we get to talk about that second R01 today. And one of the reasons that I wanted to do that, A, because it's a really cool story, and B, it kind of shows that even like early stage investigators, you can learn something new. So I was really fortunate to get one of these CATS R01s. If you haven't heard about them, you have to do something totally out of your wheelhouse, and you're not allowed to show any preliminary data. So they're really hard to review for study section, but they're actually like a great opportunity. So I'll tell you that story today. And you can hear me okay on Zoom? Thumbs up. Okay, I hope so. We're
Can
on.
someone confirm on Zoom?
Okay, we got one, yes. Thank you. Okay, so to get started, I want to acknowledge my lab, and then the second picture on the right is another person. That's, oh my God, everybody can hear now. Maybe there's a lag, I don't know. But so in my lab, Dr. Zhao Yang has done all of these analyses with the preterm birth studies in our mouse models, but I would not even have gotten into this without Dr. Gil Moore. So Gil is in the middle there. He's a well-known leader in maternal inflammation, and he actually got very famous for Zika virus. So he was one of the first people to show that a virus could impact offspring health. And he has a great lab at the Mott Center, and we actually brought him into our super fund. So that's how our relationship actually started. But I am fortunate to have both a P30 and a P42, so we have funding through them, but also the ECHO program, which is really cool, because now I can validate some of my observations in mice with these human cohorts. Okay, so what's going on in my lab? So I have a study probably at each of these time points, and they're all related to PFAS. I've gotten really interested with PFAS since coming from Kentucky, because I live in Michigan now, and Michigan is a hotspot for PFAS. And we were also told by the community that that's what they really care about. So I wanted to kind of study what's important to my community. So my first, my bread and butter is looking at adult exposures and atherosclerosis risk. So this kind of end age thing. But with this CATS award, I'm able to kind of do pregnancy studies, which I've never done before. We have some studies with Rick Pilsner, who you may know, with sperm methylation and PFAS. And I've kind of drank the Kool-Aid with the paternal exposures. Every time we do a paternal exposure, whether it's a diet study that we have coming out or a PFAS study, things are changing, especially for cardiometabolic disease. We have an interesting study with Lauren Petrick looking at ovarian aging in mice. We have some hormonal issues with PFAS. So I'm just really interested in PFAS, how can it impact across the lifespan. And one other thing here, I'm also really interested in how bugs can make things worse or how they can mediate things. What I mean is how your gut microbiome can mediate some of the outcomes, because we've shown many times that PFAS can modulate gut microbiome. Okay, so just a brief kind of like blurb about our P30s, because I think we could work together really well, because we're both urban P30 and P42 centers. We have the Center for Urban Responses to Environmental Stressors, CURES, and it's been a long-standing P30. We do a lot of the same things as you guys. So we have the things that we can't talk about anymore with health disparities and that we can say. But we're 80% African American in Detroit. So a lot of the same issues that your communities are dealing with, we are. And then our super fund is really cool because we're focused on preterm birth. We've identified that as probably the most important health outcome for Detroit, at least compared to other cities. So now we're trying to investigate the environmental impacts on that. This is the makeup of our P30, if you don't know. So it's run by Melissa Runge Morris and Christine Cole Johnson. It's been a long-standing relationship between Wayne State and Henry Ford. Henry Ford is the largest hospital system in Detroit, and it's literally right across the street. And so we have really good toxicology group through the Wayne State side, and amazing clinicians and epidemiologists on the other side. These are our research interest groups in the green bullets. We're really interested in environment, metabolic health, and all those other words with PrEP. We're interested in health, environmental awareness, and resilience. So this idea of resilience and, for example, you heard nutrition. How could nutrition make you more resilient? But then we also have a brand new rig on pregnancy and asthma that Christine Cole-Johnson is running. They have amazing biobanks and studies going on there for asthma. And then for our Superfund, we're really interested in preterm birth. And we have 3 projects as of right now. That's probably changing for the renewal. But for 3, we have a transdisciplinary approach with a zebrafish placental mammal model, which I help out with Gil. And then the Henry Ford folks focus on our human population. They built a cohort from scratch looking at preterm birth and benzene, toluene, ethylbenzene, and xylene BTEX exposure. And then we have 2 environmental science engineering projects. One is actually measuring these BTEX compounds through plant species, so a novel way to sense these things, and then also remediation and using AI and some really cool approaches there. So again, this is what we've done the past 5 years. Once the RFA finally comes out, we'll do something new, but in the same idea. Preterm birth is actually a huge issue in Detroit, and I should have looked it up for Manhattan as well, or Bronx, or these other parts of New York City. But 14.4% of live births are preterm, and the average in the U.S. could be 5 or 6%, if not lower. So it's a huge issue there, and it's even higher just in Black infants. So, you know, in school of public health, there's a lot of different things that are going on, socioeconomic, but we're really, really interested in interactions with the toxicology side of things. Because you all know Detroit, I like to say it was Chicago before Chicago was Chicago. All the industry in the Midwest was through Detroit. Obviously, you had the automobile, but other industries too. And now we have that legacy of all this crap that we have to deal with. And some groups have to deal with it more. You guys know that. And so we focused a lot on the BTEX side for the Superfund. And just because I was part of that, I got to meet Gil more and learn about preterm birth. And I'm like, hey, Gil, what about PFAS? It's important for Michigan. We don't have any idea if it's important in Detroit, but we know it's important for the rest of the state. Do you think we could develop some projects around PFAS and preterm birth? And that's what this grant came about to be. And so just one brief thing about Gil's work. He's done a lot of really great work. He talked about bringing new people into your P30. When we brought him in, this got the best score for sure for any of our projects. He's an excellent scientist, but he's never done anything environmental before. It was always infection. And he had this idea of, well, what about a viral infection and a bacterial infection? Well, what about a benzene exposure and one of these other things? And so he's come up with a nice past 5 years past literature on this. And what's one of the cool things that he taught me is that there's placental sexual dimorphism. And what I mean is a placenta that's attached to a male fetus is going to respond differently to a placenta that's attached to a female fetus. And this is something I had never really thought about. I said, oh, it's a placenta, it's feeding the baby kind of thing. But it actually responds differently. And it may be one's more pro-inflammatory versus the other sex, or with my PFAS, maybe more immunosuppressant or more metabolically different. But it's just something I think we should all think about when we're doing our human studies, for example, or our molecular studies. We have to sex the placentas if we want to get the real information. We have a bunch of PFAS projects going on, so if you're interested in getting into this realm, we probably have a health outcome that you're interested in. So you heard what I do. We have someone that's doing bone metastasis and cancer. That's Isabel Podgorski next to me. You probably have seen Rick before. He's the sperm guy. Judy Westrick is our analytical chemist. So she's getting involved in some exposomics and some PFAS measurements there. We even have a heart failure. So if you're interested in like latest stages of life, heart failure, that's what Suresh is doing. And then we even have engineers that are interested in this. So for our Superfund, we're going to look at volatile PFAS, and that's led by Carol Miller. So if you're interested, I can put you in touch with these folks. And all of these were built through our P30. That's the other thing I'll add. So thanks to the pilot program and all of that, we've been able to kind of get projects going. Okay, so to the research. I'm going to focus on the maternal side today. Okay, so preterm birth and PFAS. So the map on the right is just showing preterm birth rates throughout the world. It is an international issue. You can see some of the hotspots, and unfortunately, it doesn't hotspot Detroit because 14.5% is very similar to some of these dark purples. And so there's been lots of links between PFAS and pregnancy outcomes. I'd say preterm birth is pretty reproducible. Intrauterine growth retardation, so smaller babies, has been pretty reproducible. And anything immunosuppressant. So looking at like antibody response in children, these are all very reproducible in human cohorts. And for Michigan, like I said, it's like the most important thing that we're studying right now, because look at these concentrations. I just pulled out one example. So Kalamazoo County, if you like beer, they make some of the best beer in the country there. They have almost 6,000 parts per trillion found in wells in Kalamazoo County. We have well-known exposure routes through Wolverine manufacturing. Even in Detroit, there's known spaces. So it's really important for Detroit, but it's not, and Michigan, but not just Michigan. Like I went on the website. Supposedly, there's $2.7 to $4.4 billion spent on burden in New York just on PFAS. And so I like to pull something from whenever I give a talk. This is not just a Michigan problem. I like to say the biggest problem. It's our DDT, my generation's DDT. That's what I say. You all know probably a lot about PFAS. The only thing I really want to focus on here is they're found in complex mixtures. So this idea of studying one PFAS at a time is hard, and it's probably not giving us the full story. And so as a molecular toxicologist, that's what I always taught. You got to go down to the nitty gritty, one contaminant at a time, study dose response of one thing. I think PFAS is the perfect example where we can't do that. But then how do you weigh it? Like, okay, we're going to do mixture methods in humans. Are we going to do mixtures in mice and cells? It just gets very dirty. So we have to find a happy medium where we can study mixtures, but also kind of figure out what's really driving that mixture. So some people say 4,000 different PFAS. I put a 1 there. Maybe there's 14,000 different PFAS. There's breakdown products. There's different types. It's a huge class of compounds, and they're building more all the time. You know where they're from. And then the other thing is that most of the exposures nowadays are from ingestion. So whether that's your diet or your water, but there are recent exposures through inhalation. So whether this is through vapor intrusion or just through like stuff in the house that's like off gassing, it's definitely something that we have to study. Okay, so with those 14 possible 1,000 different PFAS, there's lots of ways to characterize them and clump them together and categorize them. And here's just 3 very easy ways to do it. You can look at the legacy compounds. So these are like those C8 long chain PFAS that they make movies and Netflix specials on. You have alternatives, so alternatives for PFOS, for example. And then you have these emerging ones. So Gen X is a very common one. We know a lot about Gen X with North Carolina. But there's lots of other ways to categorize them. So when I was thinking of how am I going to do a mixture of PFAS in mice or cells or study them in humans, this was an easy way for me to clump them together. So I wanted at least one PFAS from each of these groupings that I was going to give the mice. The other reason that I would focus on these is because we have good epidemiological data linking these to whether it's pregnancy outcomes or like cardiovascular disease that I was focusing on. So on the left there, this is the well-known C8 cohort, manufacturing in Parkersburg, West Virginia, contaminated the Ohio River. And then on the right is the well-established Chemours plant in North Carolina and Gen X. So we have really good epidemiological links with what we're studying. So what are the levels of these compounds? So if we just look at PFOS, which is the most predominant in the blood still in the United States, it depends where you are and where you work. So for example, that top one there, this was a manufacturer workers in Alabama, almost 1 part per million level of just PFOS. Now remember I said there was 14,000 of these things. So very, very high concentrations. Compared to some other contaminants that you might study that might be at the parts per billion level, these are very, very, very high. And of course, it's different for everybody, but it's higher than other contaminants. One of the reasons that they're so high is because they're long half-lives. So like PFOS has a 5-year half-life. PFHXS, which we found to be high in Michigan, is almost 10 years. So obviously this plays a role in why they're so concentrated in humans. And then on the right, there are links. This is a meta-study, a meta-analysis of 14 different papers. They found links with PFOS and preterm birth. So there are rigorous studies out there linking that to what we're studying. Just so we're all on the same page, we call preterm birth any birth occurring before 37 weeks, and it's escalating globally. So it's not a success story. It's actually getting worse. There might be up to 15 million babies that are affected by preterm birth or this related intrauterine growth retardation. And probably most importantly, 75% of perinatal mortality and 50% of neurological issues come from preterm birth. So we have to study this so we can stop all of these diseases after birth. And then I talked to Gil. He brought this to my attention, which I didn't know. I don't know if you know this. Preterm birth is associated with infection 40% of the time. And that kind of blew my mind. But then on the other side, moms are getting infections all the time. Why are we not all getting preterm birth? And so I think that's like a really important question that can kind of lead you down different directions. And so that's exactly what I have bolded here. So if and when, why does a microorganism does induce preterm birth? What are the protections that moms actually have? And it's probably at the site of the placenta. And so that's what I'm going to be focusing on for the research part here. And it's what Gil subscribes to is this idea that there's protective mediators such as interferons. So we think of interferons as bad for you in the circulation. They're actually very protective in the placenta. But if you suppress your normal interferon signaling, that's when a secondary stressor can come in and elicit a negative effect. And so what Gil has shown in mice is that if you give a LPS to a mom, nothing's going to happen. So the mom will just have the baby normally. There will be no issues. But if you had a first stressor like an infection from a virus, that's going to tamp down your interferon signaling. And then if you give the LPS, then you have, it's not preterm birth in mice, it's preterm delivery. And then like a circulating inflammatory cytokines that go really high too. So he has a lot of really good evidence for this two-hit hypothesis. But I'm the toxicologist, I was thinking maybe PFAS could act as this first hit. So maybe not the virus, but maybe PFAS, because they are well-established immunosuppressants. It's one of the most reproducible effects in humans is immunosuppression due to PFAS. And there's some examples on here. One thing that always pops up is infection severity and also antibody response, associations with that. And we know from human studies that the PFAS actually makes it to the human placenta. So that's what the little figure is. So I thought it was worth a shot. I wanted to write a grant on PFAS as the first hit for this two-hit hypothesis. So we got the grant, and so we've started doing some things with cell culture. So this is trophoblast cell line that Gilmore has developed, SWAN 71 cells. The first thing we did is we exposed them to different PFAS mixtures, and we looked with transcriptomics, and I pulled out all those interferon-related things that we were just talking about. And just like our hypothesis would expect, PFAS is immunosuppressant. It decreases response to interleukin-1 along with this type 1 interferon signaling. So it's like, oh, our hypothesis might be right. Now let's develop this mouse model. Okay, so our hypothesis, based on all this, we do propose this new double hit hypothesis where the PFAS will act at the site of placenta to downregulate interferon signaling. And then if we give an LPS or another bacterial stressor, then we'll elicit that preterm delivery and cytokine storm. And so this is what our kind of in vivo design looks like. What we do is we order mice, pregnant mice, plug-positive mice, and we don't do anything with them for 6 days. So they arrive at E2.5, embryonic day 2.5. We put them on our standardized diet, because I like this standardized diet. We can talk about it later, why I think this might actually be playing a role in what we're seeing. And then from E8.5 to the very last day of development, we give our mixture of PFAS. And it's that same mixture that I was talking about with that category. So we give PFOA, PFOS, PFNA, PFHXS, and GenX at 2 milligrams per liter. And then at the end, right before the moms give birth, we euthanize them and collect decidua, placenta, fetal membranes, all these different types of things. And we do metabolomics, spatial transcriptomics, and bulk RNA sequencing. And that's what I'll talk about today. This diet and this PFAS exposure, we've used a lot of times now for our cardiovascular endpoints. So this was the first paper that we used that specific cocktail and the diet. So if you're interested in that, you can take a look. Okay, so what do we see? So I'm going to throw the phenotype up first. We get a surprising phenotype. Remember all that talk I said about intrauterine growth retardation and small pups and preterm birth? Well, if we expose mice for these 10 days from E8.5 to 18.5, we actually get bigger male pups. And no one has really shown this yet in mouse studies. There's been some really great work from Carolina and other places where they do see this intrauterine growth retardation. But they give the PFAS throughout the whole pregnancy, and they don't give the diet that we give. So it's going to be a little tangent, but why do we do E8 to 18.5? First study we gave it from as soon as we got the mice. We got no pregnancies. So we were like, oh crap, this is impacting on implantation, a totally different project. It's a cool project, but it's not what we need. We need placentas. We need fetal pups. So we talked to Gil, and he said, well, if you wait till E8.5, the placenta's already developed in the mouse. So you're kind of studying like a second trimester susceptible window. And then as soon as we did 8.5, we got plenty of pups, and we see this offspring effect. And we've done this 5, 6, 7 different cohorts, and we always see it. And we just euthanized mice at E15.5. We also see it. So even as early as E15.5, we see an increase in pup weight in the males. Nothing with the females. And these are different ways that you can show growth in fetal pups. And this has nothing to do with the placenta, we don't think. There's no changes in placenta weight, and there was no real impacts on litter size. So we think anything that we're seeing is irrespective of these changes. And we actually measured PFAS. So if you're curious, how does that 2 mg per liter for those 10 days relate to humans? It relates really well with the placentas. So we got female and male here. There's no sex differences on the concentrations of PFAS. And they're averaging about like 2.5 nanograms per gram to like, I don't know, is that 4 for PFHXS? And it's very close to what we're seeing in the human study. So we're not hundreds of times higher. So we're really happy with this exposure. First thing we want to look at was the immunosuppressant or immunoactivation. So this is circulating cytokines of the moms. And long story short, we don't see much of anything. So there's no cytokine storm. There's no large-scale immunosuppression. The only thing that we see significant is one protein, one cytokine, and that's interleukin-17 alpha. And so I didn't know much about interleukin-17, but now I know a lot. So it's a pro-inflammatory cytokine known for autoimmune diseases. And it's primarily produced by T helper 17 cells, which is really interesting because people haven't really focused on this yet, especially in pregnancy. But along with these autoimmune diseases and preterm birth and preeclampsia, it's important for psoriasis, arthritis, and these other kind of autoimmune diseases. So something to look into further. But I think this is also important to say that any of the effects that we see downstream don't seem to be just because of a large maternal inflammation. So on one way, it stinks that there's not immunosuppression or a lot of immunoactivation. But now we can kind of look at specifically in the placenta, what's the PFAS doing, irrespective of just a cytokine storm. Okay, so what did we do? So we did a lot of histology for this study because we wanted to do spatial transcriptomics. And if you guys don't know what spatial is, it's like the coolest thing out right now. There's lots of different levels of it. So we chose Visium HD, which is kind of right in the middle, sweet spot for cost and coverage. If you want to be at the super single cell level, you want to use Xenium. But Xenium has drawbacks. You can't do a full transcriptome-wide kind of analysis. You can look at, I don't know, they keep upping their number, but it's not a full transcriptome. So Visium HD was great for us because you can do single cell transcriptomics on a tissue slice. So if you guys have a biobank of any human studies that were just normally formalin fixed and thrown in a freezer or fridge forever, you can go back and do spatial transcriptomics on them. Versus, okay, if I want to do single cell, then you have to plan that out ahead of time. This is something you can go back in time. Okay, so what do we see? First we did, before we got to the spatial, we just did bulk transcriptomics. And looking at the placenta here, one thing I want to point out, if you guys don't know, the placenta has a maternal side and a fetal side. The maternal side is called the decidua. That's what's on the right. And what was kind of interesting to us is it looks like the decidua is more susceptible to the maternal PFAS than the placenta. Now remember, this is E18.5, so it's very end, so some things at the placenta might start to shut down at this point. But even just that, like the decidua has this long-lasting effect for the bulk transcriptomics. And I'll just point it out here, in the placenta we see this fatty acid binding protein 1, because the point of doing the transcriptomics is to kind of find mechanisms that could link to your phenotype, right? This is a fatty acid binding protein. So it makes sense that, oh, maybe this is something we have to follow if we're interested in this fetal growth phenotype. If we start to look at pathways, this is where we can finally see this sexual dimorphism that might help explain maybe some of those phenotypes. So on one hand, we see very similar phenotypes between sexes or pathways with hypoxia. So that's what the green arrow here. It's the number one pathway in both male and female placenta. But then we also see very different effects with these inflammatory pathways. So, for example, in male, we see an enrichment of TNF alpha and an NF kappa B. Those are your pro-inflammatory cytokines. But in females, we see the opposite. We actually see a de-enrichment, if that's the right word, like less of that pathway, which is really interesting. Why we see opposite effects just for the inflammation. Versus the decidua, remember, it was more susceptible, but there is no sex differences here. Pretty much across the board, the pathways are exactly the same. But again, the hypoxia keeps popping up. And if I would have seen intrauterine growth retardation like everybody else, I would have pointed to hypoxia right there. That would have explained everything. But we actually see the opposite with our phenotype. So we still have less oxygen at the site of the placenta, but we still see more growth in males, which is kind of a really interesting dichotomy. These are some of the genes with the hypoxia and that you may be focusing on. A lot of these are downstream of HIF1 alpha, and I know there's some people looking at PFAS and HIF1 alpha. So we still see the induction of these genes, but it's not causing fetal growth restriction in either of the sexes. And it's probably due to our diet. I'll toss that out there now. Okay, now moving to the single cell spatial transcriptomics. If you haven't worked on this before, I think it's really cool, so I'm going to walk you through it a little bit. So what you can do is, if you've ever worked with 10x genomics before on single cell, you'll get these UMAP things on the top left. So at the single cell resolution, you can kind of figure out what's happening in that cell. And because of that, you can figure out, is a certain cell type more susceptible than other cell types to your diet or your pollutant or that kind of thing. So that's what you see on the right. So these are all the different cell types that we get picked up in our bioinformatics pipeline. And it's everything that you would hope to see. Decidua, endoderm, ethroid, fibroblast, giant cell trophoblast, glycogen trophoblast, pericyte, spongiotrophoblast, and yolk sac. So our first run bioinformatics pipeline gives us all of these cell types. So we can actually look at ratios. Does the PFAS change the ratio of any of these? And in our model, no, it doesn't. So when we look at these 11 cell types, there's no difference in the number of those or the ratio, which is cool. There's no impact on that. But when we look at the genes, there's a huge impact, cell-specific impact. And the next slide will be female, so I'm going to show the sex-specific examples again. There's a sexual dimorphism of the genes that are differentially expressed due to PFAS. So I have two starred here, the glycogen trophoblast and the spongiotrophoblast. So in male placenta, we see almost 150 significant genes for the glycogen trophoblast due to PFAS and the spongiotrophoblast. Versus, if we look at in females, we actually see more repression in the glycogen trophoblasts than in the males. So there's this, I think that's really important, because what are glycogen trophoblasts known to do? They're actually storage of glycogen, which is an energy source. Oh, so we're trying to explain that growth, the development of the male. Maybe this has something to do with it. And then with the spongiotrophoblast, remember, in the male, we see this strong red induction, repression, and then here we see it's kind of like an even. So these are two pathways. The other one that we're really interested in is in the yolk sac. So yolk sac's obviously important for energy during fetal growth. There's some really interesting sex differences that I don't have time to get into about those. The other thing that you can do with the spatial single-cell transcriptomics is now do the pathway analysis and the differentially expressed gene analysis of each of these individual cell types. So if I just pulled out those two that I starred, the glycogen trophoblasts, you can see that there's a sex difference here. So on the right, all the red are the female significant pathways, and the blue is the significant pathways just in males. And you can see there's some overlap. So JAK-STAT pathway, lactation, T-cell activation, virus, okay, that's really important, and blood coagulation. But you can see these other ones are very different, male versus female. And just pointing a couple with the male, ERAD and protein response are important. Oxidative stress of the cell is enriched for males. And then, ding, 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 placental development is only different in males. So that's only enrichment in males, glycogen trophoblasts. So we can do the same thing for the spongiotrophoblast. And here I think it's even more obvious. You see there's 4 or 5 pathways that are shared among the sexes, but it's very different for the male and the female as well. So I just think this is such a cool opportunity to go back in time, but also to do mechanistic studies really well at the single cell level. And for anyone that's doing like cancer, you can actually, on your tissue slice, pick, I only want the bioinformatics pipeline for this portion. So say your cancer portion of your cell, of your tissue slice versus the regular in one tissue slice. So it's a great technology. The other thing that you can do with it is this cool thing called cell chat. So you know that there's ligands and there's keys and locks everywhere. Things are being secreted by one cell type and autocrine function locking to another transcription factor or ligand binding. And you can actually do this with bioinformatics tools. And then you can compare PFAS versus regular. And one of the cool things is we can see a sex difference if we just look on the left side here. These are just controls. So this is female control versus male control. You can see that there's a difference in the cellular chat, the connection, the communications, just in baseline, male versus female. So irrespective of any PFAS, whether you're a male placenta or a female placenta, it's different. And then you can look at your PFAS. So at the top here, you can look at this very dark purple, thick line, for example, going from fibroblasts to the giant cell trophoblast population. You can compare that to over here, fibroblasts to giant cell. That thickness of the line is less, meaning there's less interactions between these two. And then you can look at also in your male. Here, it doesn't look like there's as strong as a communication between fibroblasts and giant cell compared to over here. And maybe it's different, maybe it's not, but there's other very obvious differences. Fibroblasts versus endoderm cells, for example, in the males. So this is something that can kind of be like hypothesis driving. So it's a cool thing. You also get just for statistics like a total number of interactions. So on the left, that's a total number for control versus total number of PFAS. I don't have it split by male or female just for space. But you can see PFAS is decreasing the number of interactions between the cells. Now, how is it doing that? We don't know. But it's definitely something to look into. The other thing that the cell chat can do is, yes, it can look at total number of interactions, but then you can actually tease out what's driving those interactions. So I have 2 examples. One example here with this galectin pathway. So in control for males, you see a strong, thick line between glycogen trophoblasts and spongiotrophoblast, but you can see obviously that has been decreased. And you've also lost these other connections, like glycogen to syncytia, trophoblast, and so forth. So then you got to think, what is this galactin pathway? It's actually related to sensing of sugars. And I didn't say it, I didn't put it in the thing, but we think that PFAS is changing the glycoproteome a lot. So we have a whole aim for this preterm birth study looking at, we call it glycoseq, using this type of sequencing technology to look at glycobiology. So it was cool to see this. Okay, one thing else you can do, so everything I've shown up here is just the first pass bioinformatics. This is what most people would do. There's another alternative way to look at the bulk transcriptome, and that's a deconvolution bioinformatics tool. And so I looked at my data and I talked to the core and I'm like, but we're missing a lot of different cell types. I only see 11. I know there's a lot of inflammatory cells, for example, in the placenta. I don't see them in the results. And there's like, well, they're low abundant cell types. We can try a deconvolution technique where we can pull out lower abundance. So I said, please do that because I'm really interested in, say, macrophages, for example, or Hofbauer cells. And so she did that. And now look what we see. We have, what is this? 30, 25 different cell types that they can find. Importantly, B cells, macrophages, neutrophils, natural killer cells, all these really important inflammatory cells we now can investigate, do everything that we just did, pathway analysis, differentially expressed genes with a lot more different cell types. And here we actually do see changes in the ratios. So once we do the deconvolution, we see interaction terms, meaning the effect of the PFAS is dependent on the sex. For deciduous cell types, endothelial cell types, stromal cell types, and these progenitor trophoblast cells, which is interesting to look into. But we wouldn't have gotten any of this information just using the normal bioinformatics pipeline. So I was interested in looking at, is there any inflammatory differences here with these cell types? And there is. So for females, we see Hofbauer cells and neutrophils being enriched, and there's lots of different pathways that are significant. We don't see this at all with the male placentas. Versus macrophages, we actually see macrophage enrichment much more in the males and pretty much nothing in the females. So there's a shift. Why are the males activating their macrophages, or why is macrophages being impacted more in males versus females? And why are these two inflammatory cell lines more enriched? A lot of things to look at. Okay, so thus far, PFAS mixture results in measurable PFAS in the placenta. That mirrors humans really, really well, so we're very happy with that. And there's not an overt maternal immune activation like we see with the benzene and Gill's project, for example. So we don't think that maternal immune activation is causing all of this. Our surprising result is that the growth and development appears accelerated, but only in the male fetuses. And this is head, this is body weight, pretty much everything we're looking at. It's increased growth. Looking at the bulk sequencing, decidua is more susceptible at this late time point, but the sexual dimorphism is more evident in the placenta. And then we think that the spongiotrophoblast and glycogen trophoblasts are like low-hanging fruit that we have to really focus on more. Okay, so what about fetal tissues? So I said we also collected as many fetal tissues as I could see. So this is, with PIs, we don't work as much in the lab. I love working with the mice, so I actually did this part myself. I'm proud of myself. So I isolated fetal brain, I isolated fetal livers, and fetal hearts. These things are really small, so I'm proud of myself. My postdocs laugh at me, but I'm proud of myself. And with that, we did transcriptomics. So we did bulk transcriptomics, no Visium HD yet. We just wanted to see, at this concentration of maternal exposure, do we see anything in the development of these important organs? And we absolutely do. So this is just a quick summary for time. For the brain, we see 817 differentially expressed genes in males and only 200 and some in females. In liver, we don't see much of a sex difference, but look how many differentially expressed genes. So fetal liver is a really important target for maternal PFAS. And then the heart as well is almost double male to female. So again, this is a very short exposure. This is only 10 days, but actually a really important time of development. So maybe that's the reason that we're seeing such an effect. If we look at the liver, remember, we don't see much of a sex difference here, but we see many, many genes related to lipid metabolism. And so your favorite lipid metabolism gene might be up there, the ACOTs, ACOXs, all these things impact on lipid metabolism. And for me, someone that's interested in cardiometabolic disease, this is perfect. Now I need these mice to be born and to challenge them with high-fat diets and Western diets and everything and see if they're predisposed to the diseases that I know best. But no sex difference. We looked at, this is transcription factor binding with a bioinformatics tool. And this is the first time that we see what everyone always sees, is PPAR alpha. We did this transcription factor binding assay for all of our tissues. We never saw PPAR alpha pop up. Decidua, placenta, any of that stuff. We see it in the fetal livers though. And it's the number one transcription factor that's enriched. And for anyone that's studying PFAS, you would expect this. This is known in mice that it activates PPAR, especially PPAR alpha. But I thought it was super interesting that we're only seeing it in the liver. And if you go back to our volcano plot here, a lot of these things that are induced on the right, they're downstream of PPAR alpha. So we're seeing probably a PPAR alpha effect here, but not in the placenta or decidua. And yes, we measured PFAS in the fetal livers too. So we just got this data like 2 days ago, so I put it up here. Interestingly, we see more PFAS in the fetal liver than we see in the placenta. And this is not what we see in humans. So I found one paper that looked at fetal livers in humans, and it's about one-fold decrease concentration compared to the placentas. So like PFOS 0.2 to 4, we're at, 10 nanograms per gram. But okay, the numbers are close, but what I'm saying is our placentas, we had 2. So we actually see more in the fetal livers compared to placentas, and in humans, it's flipped. They see more in the placenta than in the fetal liver. And we don't know what's driving that change in the ratio. Maybe it's the diet. So I've said the diet a bunch of times. So the diet that we give is a low-fat diet, so that's not impacting it, but it is enriched for cholesterol. So we really think that cholesterol can help transport these PFAS, and we are always interested in for the cardiovascular hyperlipidemia outcomes, but it makes sense too. It could transport across the placenta more because the PFAS can actually be bound and transported by these things. We have a really interesting human study where it was a lipid lowering study where we measured PFAS pre and post intervention in humans, and we actually saw, yes, PFAS went down, but more interestingly, it changed where that PFAS was located. So like VLDL, LDL, HDL, it changed where it was sequestered. So we think the high cholesterol in our diet is playing a role here. Okay, I think this might be the last figure here. So I'm like, okay, Zhao, I want to know as much as we can about things related to nutrition. So I want to try to explain this fetal growth phenotype as best as possible. So I want to look at genes known to, related to amino acid metabolism, fatty acid metabolism, glucose metabolism. And what he sent me this, and I'm like, really? That's it? So we see no changes in any of the glucose. So our fetal growth has nothing to do with changing metabolism of glucose. At the top, we see nothing with amino acids, but we see a lot with this fatty acid binding protein and fatty acid related metabolism. And across the board, we see this FABP1, whether it's placentas, livers, brains, hearts, it's always upregulated and induced. And it's really important for bringing lipids into the cell. So we think this is obviously, we have to tease it out more. We can do some cell culture stuff. Maybe there's some inhibitor studies we can do in future. But when you see a heat map like this, you have to focus on that. Okay, summary. So maternal exposure to PFAS increased the fetal growth just in males. And we think that there is an interesting maternal diet and toxicology interaction here. And we're going to actually rigorously test this now. So we're going to just use, I'm not a big fan of chow diet. So if you do mouse studies, I don't like chow diets. I like standardized diets. So we'll do a standardized diet that matches what we have here, just regular cholesterol, and see if that impacts anything. And now we're also going to do our two-head hypothesis. So that's what we're starting Right now, we're using this model, and then we're going to give LPS at the very end and see if we see that Gilmore effect. So stay tuned. So I'm at 10:46. Is it a good time to stop? I have one little study that I would love to tell you about. Just 5 minutes? But yeah, so if you're interested in our Superfund and our cures, here are our websites. Matt already told me he loved my website, so you can check that out too. I didn't make it. But so I came here. We might have some good news about this study to talk about for funding. But what about the dad? So we've gotten so heavy into the mom. Dads are exposed to the same things. A lot of times they're in the same neighborhoods, the same households. What about paternal exposures? So just like 3 or 4 minutes, I really want to talk about this because I think at the end you'll see it might be linked to what I was just showing in the placenta. So this was a P30 initiated collaboration pilot project. Our pilot projects are rarely large, so we actually do $65,000 pilot projects. And because we give that much money out, we can actually do these type of really actual studies, which is cool. So we did one with Rick Pilsner. He came from Amherst. He had gotten famous, I'll say famous with quotes because it's Rick, about sperm and phthalate exposure. Yep, we talked about that. So he was at University of Michigan too, so he did some of that at Amherst. And when he came to Wayne State, I'm like, oh, would you like to collaborate at all? And he said, well, what do you do with your sperm? I'm like, I don't do anything with testes. So we actually got one of these grants, and then he did DNA methylation, multiple ways. We've done like 5 different projects now with him. Every time I do something with the paternal exposure, something changes in the offspring. And so we got a really cool paper out in Environment International about this. We measured these different, we used the same PFAS at much lower concentration because we wanted to do a long 18-week study in the males because with like the sperm, it's like a 30-day turnover for sperm. So this is about more than 2 or 3 spermatogenic cycles. We can measure all but the Gen X in the testes, which was cool. This actually came out very similar times as that nanoparticle, microplastics paper with like the placenta. So we were like competing PFAS in the testes versus plastics in the placenta. And then he did his methylation, and you can just look at the colors, PFAS versus control. The methylation in the sperm is very different. And so then the question is why? And then that's what you get a grant for to do. But more importantly for me, it's like, okay, you're showing sperm is different. What about the offspring? Are they predisposed to cardiometabolic disease? And so in that same paper, we did that. And it was interestingly, we did see a sex difference here. So these are offspring from the dads that were exposed to PFAS. Males, we see 40 hepatic genes that were differentially expressed versus only 9 in females. And we looked at cholesterol, we looked at other lipids, only significant in males. So another example of males might be more susceptible. And then here are some of the other lipids that we looked at. Okay, how does it get back to the placenta? So this is something I learned, and I hope our reviewers and study section people understand too. Most of the genes for the placenta are from the dad. Yeah, I'll say that again. Most of the genes that are turned on in a placenta are from a father. So this is a very interesting possible sexual dimorphism reasoning for why we might see different effects for the paternal exposures. And going back, remember we saw changes in glycogen trophoblasts. In the middle here, this was a glycogen analysis of placentas from dads that were exposed to PFAS versus dads that were exposed to just water. You can see glycogen is much, much higher in just the males. Male placentas. So we're seeing the glycogen trophoblasts enriched in our maternal exposures. We're seeing glycogen much higher, again, only in males in both of their studies. I thought it was a cool kind of crosstalk. Okay, I'll take some questions. If you have anything on the Zoom, you can write it in the chat.
I'm going to stop sharing so we can see who's writing on this one.
Thank you, Nancy.
Thank you. This was like really interesting and cool work. Selfishly, I I'm trying to model a mixture of phthalates and like parabens and phenols in a placental cellular model.
Okay.
And I'm wondering if you could talk about how you decided on the dosing of your mixture of components.
Mhm.
And it looks like it's they were at equal concentrations in the mixture. And if you could talk about why you made that decision.
Yep. So we developed this mixture when I was at Kentucky my last year, and we just did the easiest way: 2 mgs, 2 mgs, 2 mgs, equal molar concentrations. For the grant, we actually proposed a mixture that's kind of environmentally related. So the way we did that is we looked at NIEHS data, and there's a ratio in NIEHS, PFOS to PFOA to PFHXS, and then Gen X we said 1% because it's very, very low concentrations. So that's what we're going to do next is this environmental. But the reviewers were okay with just doing the simple mixture to start. And we picked those 5. Like I said, it was each one of those represented those major classes, and they all had epidemiological links to my main endpoint, which was cardiovascular disease. And it worked, so we just kept using it.
Thank you.
Good luck with your project. Anything on Zoom? Yes, Sam.
Thank you so much. It's always such a pleasure to listen to you. I always learn so much through the years. Seeing you as a trainee, the Superfund Research Program at Kentucky such a long time ago, and then seeing your progress.
It's not that long ago. I'm not that old.
I know. I know. It's actually remarkable how fast you've done your career. It's very, very good. And very interesting data, very kind of thought-provoking in so many ways. And it's great to also to see that so many times we get the unexpected and not what we are really thinking.
Mm-hmm.
But I wanted to see, if you go back to human studies, like have you been able to see maybe that sometimes things are hidden in the data? Or they look one way, but then when you are able to see things that maybe were not happening before. And then also, like in your offspring results, do you see any, like in those liver, you mentioned is there a hint that yes, this could be cardiometabolically, like this early programming that could be actually be, like big doesn't necessarily mean better in that way.
It's very true, yeah.
And so that, do you see any hints of that?
So for the first part, I've never used AI before, but I actually put that in a chatbot. Has anyone seen growth induction or increased growth due to maternal PFAS? And there are a couple papers out there. And what's kind of interesting about them is they all used mixture methods to identify these effects. So yes, for most epidemiologic studies, it's down less growth, looking at one PFAS at a time. But there are a couple papers, some in China and some in Sweden and other places where they actually see this increase. So that'll be in our discussion section for sure. The other thing that people see a lot is growth trajectories are increased due to maternal exposure. So, for example, they'll measure PFAS in moms in first, second, or third trimester, and then look at how quickly that baby actually grows. They'll look at adiposity, they'll look at growth trajectories, which is weight. And that actually seems to be increased. So we're seeing that in utero, but people do see that once the baby's born in humans. So this catch-up idea. Yeah. So people see that. And then for the liver side, we haven't dug too deep beyond that PPAR alpha side of things. I asked postdoc to do some other staining for lipids and that kind of stuff, but we don't have a good answer yet.
I haven't
A great presentation. I'm really interested in this 2-hit model with the preterm pregnancy. So you're going to look at LPS as your second hit. From the epidemiology, is it clear that it's bacterial infection, or it could be viral as well?
I think they could look at both. So most of it, again, it's vaginal infections. That's what causes it. So we're not doing that. We're just doing a systemic. Along with the PFAS or LPS, we're next going to do infection of it's called MH65. It's a airways exposure for a mouse. So we could do Zika, we could do influenza with Gill, but that's probably too much for IACUC amendments at this point. Thanks for coming.
But I have the other question I have. I found it like kind of fascinating and dramatic that you have no offspring, you have no birth
Yeah.
with the exposure throughout, because that mimics the real human exposure.
Yeah.
Somebody wants to contribute to the question.
So
And
yeah,
so can you tell us
We-
more about that?
we
Because-
talked to Gil first because we meet every Wednesday at his lab meeting, and we brought this up, and I'm like, what am I going to do? I need for the whole study, I need fetuses, I need placentas, and we've only done it one time, so I'm not going to say that it's definitely a impact on implantation or impact on, but we don't see any hemorrhage. So in that even that first study, we didn't see hemorrhages. So we didn't see like spontaneous abortion evidence at that first. So we have to follow up on it, but it was kind of like a striking effect. Plenty in the controls and just nothing in
This is something
PFAS.
like important that we try to understand.
Yeah, and so with Gil, he leads the MOT Center. So there's people that are looking at that kind of stuff, like early implantation and embryogenesis. So we can work with them.
It seems you got a question in the chat.
Okay. Oh, I have to do
In the
it.
chat, if you go to the chat.
Airy? Okay. Oh, how changes in weather and temperature increase may change infection dynamics and relationship with PFAS exposure. No, we've not done anything related to, we can't say climate change, but this type of high temperature. I think it would definitely impact on the prevalence of the vectors and for the actual infection. I don't know how it would impact on PFAS toxicity though. So there might be people out there doing mouse studies at high temperature or low temperature, but sorry, Ari, don't have a good answer for you. But I know that is something that NIH is still funding, the high temperature, extreme weather events.
I was yesterday at the NIH, and yes,
Yeah, I think it's being encouraged.
Yeah.
they are very interested. And it seems to be part of the program.
Cool. Yeah, so you just need to get a big cohort study that's also measuring PFAS. That'd be really cool. Okay.
Any questions?
Thank you guys for coming.
Presentation.
Thank you.
I just need to.